Hello everyone, and it's my pleasure to be with you today at the Analog Astro Conference. At the first place, I would like to thank organizers for having me at such a wonderful and prospectful event. My name is Marzena Krzek. I'm a PhD in biochemistry. My main expertise focus on enzymology, including enzymes engineering towards obtaining certain and needed required functions and enzyme stability. And my second quite deep expertise is related with protein self-assembly, which I've developed at the Vrije Universiteit Brussels. Currently, I'm a founder and CEO of the see-through startup focused on biosensing solutions. Today, I'm gonna talk about my research and findouts of a very interesting experiment which I performed as a communication officer at the Lunar Mission AT at the Analog Astronaut Training Center. Just to get a little closer towards our today subject, I can imagine that some of you did not hear a lot about protein fibrillation, but all of us did hear about Alzheimer's diseases, Parkinson's diseases, or the second type of diabetics. All of these pathological states, they involved protein fibrillation in vivo in human body. And the fibrils that are formed, they are believed to be toxic enough and causing the diseases. On the other hand, protein fibrillation can assist in regular healthy physiological processes, including bone mineralization or tooth mineralization, for which fibrillar scaffold, here you can see a very schematic of a protein fibril example, and onto this scaffold a mineralization happens. So our bone, very rich with calcium minerals, is being formed. The same happens with the eggshells or seashells formation, other shells. Going further, fibrillation is also important part, or actually detrimental part, in biofilms formation. So now we move from human body and higher organisms into microbes. Microbes, they form consortia, and these consortia can form a biofilm. Biofilm causes biofolding, so sometimes we can see something is decomposing, going back, and it's due to bacterial activity which has formed a biofilm on the surface. It can happen on the surface of a spacecraft that is sent out into the space, which we absolutely want to prevent. And what's the role of protein fibrillation? When such a microbial consortium is being formed, cells do release into extracellular space proteins that do fibrilize. Such protein fibrils, they assist in cells adhesions one into another, and they do assist in the adhesion onto the surface. So when a biofilm is growing, the crucial factor is a protein fibril. Protein fibril networks within the biofilm structure also protects it from the external factors. It's been many studies described and performed taking into consideration many different factors that contribute to the final outcome of the fibrillation, but quite few studies has been focused on how incorporation of a DNA or DNA assistant for the fibrillar formation results in the final biofilm. It is relevant to study DNA because it's an abundant molecule in all described here examples for the fibrillization, both in vivo and in vitro. But next to biologically relevant systems, we cannot omit very important and maybe the most crucial for space biology, bioengineering, which involves natural materials like fibrous to form very relevant for space exploration structures. First, let's start with some examples already described and incorporated into the industry and the real life, where protein fibers are used as a scaffold for 3D printing of cultures, of cell cultures. Next to it, fibers can be used to do mineralization, same as it happens in a human body for the bonds formation, but it can also happen artificially triggered and planned by scientists. Next example are current conductive nanowires based on the protein fibers. If we will form protein fibers and later we dope them or covalently bind to nanoparticles, especially metallic nanoparticles, they do have electric current conductance. So they can serve as electric wires. They can constitute a basement and a basic structure for construction of more complex electrical tools, and maybe in the future they will be engineered and optimized for energy storage as well, which I really believe. So it's also being broadly investigated how proteins fibers that do allow for gel formation and foams formation can be incorporated into food industry for the structure of the food. And being in a space, we need to have a solution available on the site. We cannot bring everything with us from the Earth. And including protein fibers into our technology will allow having this solution being performed on a site. Now let's take a look how protein fiberization happens. I'm focusing here on structured protein or structured peptide which undergoes fibrillization. First, most important difference to understand between fibrillization and crystallization is that during the crystallization, such a native structure of protein or peptide is untouched and it just assembles into a crystal. While for the fibrillization, such a structure needs to a little bit loose up and reorganized into beta sheet richer structure later to allow ordered alignment into the fibers. Fibers, in contrast to crystals, they are aligned and organized along one axis. However, crystals are ordered and organized along 3 axes, so we can say that the fibers are 1D and the crystals are 3D. In here you can see 2 most prominent pathways for the fibrillization. The upper one is resulting in braided elastic fibers with the intermediates of oligomers, while the second one, which is very apparent to have a high content of beta sheets, much higher than the top one, have rod-like protofibers, already stiff ones, intermediates, and stiff, rigid, straight fibers that are very often confirmed to the amyloid fibers. how it is, and there are factors that have been very extensively studied so far in terms of protein fibrillization. We need these external factors in order to form and assist in protein initial unfolding, so that the structure was later organized into beta sheet and formed the fibrils. But having these external destabilization factors, we need to keep in mind that if they will be exaggerated, a complete unfolding of protein may happen, and aggregation towards random structures happens next. This process is very rapid. When you, for example, fry your egg, all the proteins in there, due to too heavy temperature jump, will rapidly lose the structure and turn white, as you could see on your fire pan. This can also be observed if you rapidly denature proteins in your in vitro study. You see rapid precipitation of a white substance on the bottom of your vial. In our studies presented here, we focused on two factors: the DNA presence, as mentioned before, why it's important, and we looked at simulated microgravity conditions, which we obtained by using RPM machine, as presented here. For our studies, we used insulin human analog, the LISPRO, which has been kindly provided by Industrial Chemistry Institute of Łukasiewicz Network, for which I thank very much. The DNA, our model DNA molecule, has been PET28 plasmid, and the results we've obtained, they were very interesting and already showing apparent differences between applied conditions, and with the strong highlight at the gravitational level conditions. So the microgravity, when you look immediately at the very first scheme showing the results, has been pretty detrimental factor in the protein fibrillization selection pathway. Here we have studied two solutions of same organized peptide, structured peptide, which is insulin. One of it, the blue one, has been a solution with the starting conditions of pure monomers, with all possible protein clusters and nuclei removed and immediately exposed to the factors we've been studying, so DNA presence and microgravity. The second one we studied has been same solution, but with a thermal pretreatment. So in here we can imagine that some initial nucleation or protein clusters could have happened, and such a solution with thermal destabilization pretreatment has been exposed to our studied factors. So these are blue and these are red solution, and let's go into details now. We looked at the microscopic images of atomic force microscopy of the endpoint samples. From the microgravity, both samples in the DNA presence and absence, they had fibril structures. And it was in line with macroscopic observations where the turbidity increased and liquidity decreased, so some gelification could have happened. It's in contrast with the results obtained for the ground control, so 1G, where in presence of DNA, fibrils were formed. Same observation as for the microgravity for the vial, while without the DNA in conditions 4, no apparent changes were observed in the vial, and oligomers were being detected by the AFM. So what we have to understand about fibrilizations is that structural reorganization into beta sheets takes time, and alignment of single molecules one next to another when assisted with some scaffold goes easier and faster. So what could have happened here is that normally such a solution on the earth in the earth conditions forms oligomers, and then the oligomers can be both with the organized structured and reorganization into beta sheets, or it can be more disorganized. Surely they were soluble because we couldn't see any changes macroscopically. While into such a conditions we add DNA, the DNA assists in the alignment of the fibrils, and this is enough to allow for the fibrillization. If we transfer the same solution to the microgravity when the mass transfer is impaired and everything slows down, we do have our fibrils formed. So we can say that microgravity facilitates fibril formation. And these fibrils in condition 1, condition 2, condition 3, they had exactly the same structure. So looking at the cross-section profile was a little bit triangle-like for both samples for the microgravity. So the microgravity assisted in certain morphology, while the fibrils formed with the assistance of DNA in a ground control conditions had a different cross-section, which you can see here is more flat. So DNA presence in this study was a predominant factor for the final morphology of the fibrils. It's important to understand that the morphology is related with cytotoxicity. Now let's see another solution of ours after initial temperature elevation, so temperature pretreatment. Here the only observed factor for the selection of aggregation of proteins was the gravitational level. Samples from microgravity did organize into rigid, stiff fibrils, probably amyloid fibrils, which has been further confirmed with the THT assay as certain dye that binds to beta sheets. In contrast to the samples that were exposed to gravitational level, no matter the DNA presence, where heavily disorganized precipitation happened. It was also manifested by macroscopic observations, as you can see on the picture here. Ground control samples, the two here, have white precipitates of proteins. It's disorganized, no structure, no fibrils, while in the microgravity, jellification happened, turbidity increased, and fibrils are observed with high content of the beta sheet. In conclusion, our studies has shown how detrimental the alteration of gravitational level can be for the selection of protein aggregation pathways. We studied microgravity, and we have found out that no matter if we take a solution pretreated before, so probably prenucleated, or monomeric solution, the exposition to microgravity will facilitate fibrillization, which is, in our understanding, mostly caused by the slowing down of mass transfer, and this time given by impart mass transfer allows for more ordered structurization and reorganization into fibrils, not only at the secondary structure level, but also at the elongation level. All these data are important for future understanding of protein fibrillization, relevant physiological processes involved in disease etiology, as well as regular physiological processes, and future bioengineering with the emphasis of space relevance biotechnological solutions. I would like to thank you for your attention, and I would like to thank co-authors of this work, Dr. Tomasz Zakowski, Dr. Anna Adamczyk, and Dr. Agata Kołodziejczyk. I'm open and happy for all your questions now.